All right, so we are going to do part 2 of the genetics lab. On the last one, we did monohybrid crosses, we did codominance, and we did incomplete dominance. In this activity, we are going to do dihybrid crosses, which means we're going to be looking at two alleles at the same time. And then we're going to be doing a sex-linked inheritance relating to the sex chromosomes X and Y. And then we will lastly do a multiple allele example. And then you're going to get some practice when you do your problem set. So again, like last time, since this is on a doc cam, the quality is not the best. So follow along on your own paper or your own document that you have on your computer that I've shared in the assignment so that you can read it to yourself better than you can see it on screen. So as an introduction, another type of experiment Mendel performed on pea plants tested the inheritance of two traits at the same time. This is called a two-factor cross or a dihybrid cross. You'll note that I wrote a 4x4 over here. We know that for a monohybrid cross, you're doing a 2x2. But in a dihybrid cross, you'll be doing a 4x4, which means that we'll have 16 outcomes, which is a lot to take care of and a lot to fit into a small space. So keep in mind that when you do dihybrid crosses, you're going to want to draw your punnett squares a lot larger so that you have room to write inside of them. In one test, he crossed a pea plant that produced round yellow peas, capital R, capital Y, capital Y, with a plant that produced wrinkled green peas. So it's lowercase r, lowercase y, lowercase y. Mendel already knew that yellow was dominant over green and that round peas were dominant over wrinkled peas. As expected, the F1 generation produced all round yellow peas, heterozygous for both the R gene and the Y gene. But what happened in the F2 generation? When Mendel allowed the F1 plants to self-pollinate, the F2 generation had a phenotypic ratio of 9:3:3:1. That is, 9 that were round and yellow, 3 that were wrinkled and yellow, 3 that were round and green, and then 1 that was wrinkled and green as a ratio. Obviously, these aren't exact numbers. Mendel could explain these results if each F1 plant produced 4 types of gametes: capital R, capital Y, capital R, lowercase r, capital Y, and lowercase r, lowercase y, as shown in figure 10.1. So the materials that we're going to use as this example are these pipe cleaners with one bead on top of them that represent a gene or by the specific color on the gene. Okay, so you will again be asked to perform crosses using colored pop beads to represent different alleles of two different genes. You will include your alleles as part of an entire chromosome to keep your genes straight. Similar to the mitosis and meiosis lab, construct two homologous pairs of chromosomes. One pair should be noticeably shorter than the other. Each chromosome of the homologous pair will carry a unique allele for a particular gene, and that allele will be represented by a different color. So I have set that up and I have created two separate ones indicating two different parents. Remember that the chromosomes you just created are present in just one individual. So now you and your lab partner are going to mate. So I'm going to be doing this all as one. So I have a red set and a blue set to represent one parent and a separate parent. And these two parents are going to mate and their chromosomes are going to be put together in random combinations. This mating may be written as capital T, lowercase t, capital H, lowercase h, crossed with the exact same. So two heterozygotes for two alleles. So for the procedure, take 4 chromosomes and mix them around on the table. Randomly select one long and one short sister chromatid. Your partner should do the same. So I'm going to do that once with the red set and once with the blue set. Okay. Keep in mind that it is important to recognize that over here we have blue is dominant capital T, so we're going to want to write that down. Capital T. We have green is recessive T, which is lowercase t. And then we have orange is dominant H, so capital H. And then purple is going to be recessive for H, so lowercase h. And that's what they're going to represent. So I want to write them down on this page so that we wouldn't be mixing up anything. So I'll leave it like here. And so basically what I'm taking is this, for one set, I'm going to be randomly picking out one long one and one short one. And what I've randomly picked out is going to be For the combination of alleles for this one gamete is going to be blue, which is capital T, and purple, which is lowercase h. Okay, so we're going to keep that and put that aside. We will be using again, and I have to do the same with the other parent to pretend like I have a lab partner, and I'm going to randomly pick one long and one short. And oh, just so happens that they're the same. So we have a blue and a purple. So it's going to be blue is capital T, and purple is lowercase h. So now record the genotype of first offspring if these two gametes were used for fertilization. So if we fertilize and we got these two as the same chromosome and these two as the same chromosome, then we're going to have capital T, lowercase h, lowercase h. Okay. Take back your chromatid so that you have the same genotype starting off for each parent. And then we're going to do the same thing for 3 alleles. So I'll do 3 separate alleles for the red, and then I'll do 3 separate alleles for the blue. And then underneath for part C here, we're going to combine them all. So for the red, our first is going to be green, which is lowercase t, and orange, which is capital H. We have green for the large chromosome, and then purple for the small chromosome. So green is lowercase t, and then purple is lowercase h. And then the last one that we're going to make is going to be, we're going to have blue, which is capital T, and then orange, which is capital H. And now we're going to do the same for the blue parent. And we're going to pick randomly one short and one long. And here we have a long green, which is going to be lowercase t, a short orange, which is a capital H. Go back into the mix. We have a long green and a short purple, a long green, which means lowercase t, and a purple, which is a lowercase h. And then lastly, we are going to pick another long and short. We're going to have blue and green, which is capital T, and then, not green, orange, and we're going to have capital H. Wow. We just so happen to have the exact same one for the both parents, which is kind of interesting. So now we are going to record the genotype of first offspring if these two gametes were used for fertilization. So we're going to look here for space 1. We're going to look at 1 and 1 here. And if you look at 1 and 1 here, we're going to have lowercase t, capital H, capital H. We're going to look at 2 now. So we're going to have lowercase t, and then lowercase h, lowercase h. And then lastly for 3, we're going to have 2 capital T's from here and here, and then we're going to have 2 capital H's. So these are the offspring crosses created from gametes created from one parent and another parent combined together to create the offspring. Okay, so for the practice problems, we're going to do tongue rolling and widow's peak, as is right here. So just a reminder, the ability to curl your tongue up on the sides is capital T. That's dominant to not being able to roll your tongue, which is going to be lowercase t. And the widow's peak, H, is dominant to straight hairline. So again, that's your the center of your hairline pointing down a little bit, and that's a widow's peak. An individual is heterozygous for both tongue rolling and widow's peak. What is the genotype of such an individual? So heterozygous means that they have one dominant and one recessive, which means that for tongue rolling, they have a capital and a lowercase, and that for a widow's peak, they have a capital and a lowercase, right? So they would be heterozygous for both if they have capital and lowercase. What are the possible genotypes of that individual's gametes? Now, here is what we're going to be practicing for the first time, and it's different from the monohybrid cross because that one you just went, if it was just T and capital T, lowercase t, you just went, oh, it's either capital T or lowercase t. But when you have 2 alleles at the same time, you have to have 2 letters. So you have to have one T and one H. So we want to cover all of the possible combinations that these could be taken together. And we're going to use the same thing that you do in math, which is the FOIL method, right? So right here, I've written it down, but I'm going to verbally go over it because it's kind of hard to see. So we will use FOIL, which means that we're going to be doing first, outside, inside, and last. You have 4 letters for this genotype. So if you do first, you're going to take the first letter of the T and the first letter of the H. In this case, it's a capital T and a capital H. And that's going to be 1 gamete. Outside is going to be the first T and the last H. So they're the outsides of this genotype. And so you would have capital T and lowercase h. Inside would be the 2 letters that are touching each other on the center, which would be the lowercase t and the capital H. So that provides a gamete of lowercase t, capital H. And then last would be the last T and the last H, which would be lowercase t and lowercase h. And this is how you get your 4 gametes that you're going to be putting across the top of your punnett square and across the side of your punnett square, given that if both crosses are heterozygous. So the possible genotypes for the gametes are going to be capital T, capital H is 1, capital T, lowercase h is 1, lowercase t, capital H is 1, and the last one is lowercase t, lowercase h. And you'll kind of get used to knowing these offhand too, but the idea behind finding them is using the FOIL method like you would in multiplication, okay? So 2 individuals heterozygous for both tongue rolling and widow's peak have children. You can set up a punnett square to do dihybrid crosses just as you did with monohybrid problems. However, depending on the parental genotypes, the square may have as many as 16 boxes rather than just 4. Insert the possible gametes from one parent in the top circles and the gamete genotypes of the other parent in the circles to the left of the box. So unfortunately, it's on the back of this page, so it's a little bit hard for us to see at the same time, but you're going to want to put the gametes that we just found here, using the FOIL method here, into the top and the side, because remember, both the parents are heterozygous for both tongue rolling and widow's peak, okay? That means that they have the genotypes capital T, lowercase t, capital H, lowercase h, and we found their gametes through the FOIL method. So the first gamete we're going to put down is capital T, capital H, and you want to make it really clear and as big as you can. The second gamete we found is capital T, lowercase h. The third gamete we found is lowercase t, capital H. And the last gamete that we're looking at is going to be lowercase t, lowercase h. And the reason why finding these is super important is because if you have 2 alleles, or sorry, 2 genes, and they have 2 alleles each, you can't have more than 2 in here in each of the gametes, because then when you add 2 gametes together, you're going to have more than 4 letters total. You also can't have 2 T's or 2 H's in here, because that means that they're going to be lacking a whole gene and having way too many alleles of another gene, right? They want to have 1 allele of one gene and 1 allele of another gene, which is why you have to make sure all of your gametes have 1 of each letter, okay? And we have the exact same thing over here because both the parents are heterozygous. So we can just copy over from here down to here. So we're going to capital T, capital H, capital T, lowercase h, lowercase t, capital H, and lowercase t, lowercase h. You can't really see it, but I just copied these over right here. And now we're going to do our typical crosses. There's just 16 boxes, but the rules are the same. So you're going to pull down from here and here. So here we're going to have capital T, capital T, capital H, capital H. And like I said, space becomes an issue when you do these. So usually having them be a little flatter is fine as long as it's longer, because you take more space writing horizontally. So here we're going to pull these 2, and we're going to have capital T, capital H, lowercase h. Here we're going to pull these 2, and we're going to have capital T, lowercase t, capital H. And lastly, we're going to pull these 2, which is going to be capital T, lowercase t, capital H, lowercase h. And I'm just going to finish these out, and you do them as well. So once you are done, you want to make sure that everything is correct. So you're going to double check everything once to make sure that you don't have any capitals where they're not supposed to be and lower cases where they're not supposed to be, right? For example, if you went here and here, you want to make sure that this box doesn't have any capitals because both gametes are completely lowercase for their alleles. So what are the possible genotypes of the F1 progeny? Well, we can see there are a lot of different ones, right? So we can just list them out. Let's list them out over here since there's plenty of space and the lines are a little bit iffy to see from. So first one that we see is capital T, capital T, capital H, capital H. Here it's a little different. It's capital T, capital T, capital H, lowercase h. This one is different as well. Capital T, lowercase t, capital H, capital H. Here is capital T, lowercase t, capital H, lowercase h. Here we have capital T, capital H, lowercase h, but we already have that here, so we're not going to pay attention to that one. Here we have capital T, lowercase h, which is new. Here we have capital T, lowercase t, capital H, lowercase h, which we already have here. Here we have capital T, lowercase h, which is new. Here we have capital T, lowercase t, capital H, which is here, so we've already gotten that. Capital T, lowercase t, capital H, lowercase h, which we already have here. Lowercase t, capital H, which is brand new, so we're going to write that down. And here we have lowercase t, sorry, I'll just say small t, big H, little h. So small t, big H, little h. Okay, we have big T, big H, little h, which we have big T, little t, big H, big T, little t, big H, little h. So we're good here. Big T, little h, little h. So we have big T, little, big, little. We have little, big, little. And then the last one we have is little, which is little t, little h, little h. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 9 different genotypes, and that is your answer to part B right here. So if you can't fit it right here on the line, that's fine as long as you have it listed out over here. Okay. What is the ratio of the genotypes now? We want to look at the numbers of how many there are, right? So the easy way to do this is after you've listed them all out and you're counting the number of how many each of them, you can just cross out with a slash because after you slash once, you can still see what is in your table. So what is the ratio of the genotypes? How many big T, big H's are there? Well, we see 1. And then I don't think we see any more. So we're going to write a 1 in front here. Big T, big H, little h. We see 1, 2. Big T, big H. Well, we see 1 here, 2, and that seems to be about it. So now we're going to be looking at big T, big H, little h. Big T, big H, little h. 1, 2, 3, 4. Okay, now we have big T, little h, which is over here. We have 1. We have big T, little h. So big T, little h is 1, 2. We have little t, big H, which is 1. Little t, big H, which is 2. And then lastly, we have little t, H, which is 1. And so our ratio would be putting these numbers next to each other, which is going to be very cramped, but we can try to fit it in here. So it would be 1, big T, big H, colon, 2, big T, big H, colon, 2, big T, big H, colon, 4, big T, big H, colon, 1, big T, little h, colon, 2, big T, little h, colon, 1, little t, big H, colon 2, little t, big H, and lastly, colon 1, little t, little h. So now we get into the last question, which is going to be, what is the ratio of the phenotypes? And the reason why I'm having you do these three things is because most questions that are going to ask you about a dihybrid cross are going to have you do the cross, and then they're going to ask you one or two of these three types of questions. So what is the ratio of the phenotypes? Well, this comes to be kind of important because the phenotypes are based on dominance and recessiveness, right? So we're going to see right here that this is dominant for both, right? So this basically is tongue rolling and has a widow's peak. This one is tongue rolling and has a widow's peak because this capital H is dominant to this lowercase h. This one's still tongue rolling and widow's peak because this little t is recessive to this dominant capital T for tongue rolling. This one is also dominant for both. Okay, after these four, the other ones are all dominant and recessive for one or the other, and then there is one last here that is recessive for both. Okay, so you got 4 plus 2 plus 2 plus 1. That is going to be total as 9. And this 9 is going to be, let me write it over here, tongue rolling and widow's peak. And so that's dominant for both. Okay, so now we're going to look at which ones are dominant for 1, which one are dominant for 0. So this one is capital T, lowercase h. This one is capital T, lowercase h. Now these two have the same phenotype because they're both recessive, so no widow's peak, but they're both still tongue rolling. So 2 plus 1 is 3, and these ones are going to be tongue rolling, but no widow's peak. These ones over here are lowercase t for both, so they're recessive for both little t's, but they both have at least one capital H. So 2 plus 1, these are going to be 3, and these are going to be not tongue rolling, but widow's peak is still there. And then lastly, we have this last one lonesome, and this one lonesome is no, not tongue rolling, and no widow's peak. So this one is recessive for both. Okay, so we're going to write this out. And this one turns out to be almost an essay when you write it out. Okay, so it's going to be 9 that are, we're going to write rolling and rolling plus peak. And then we're going to have colon, and we're going to have 3 rolling, but no peak, colon. We have 3 more that are not rolling, but peak plus peak. And then colon 1 that is not rolling and no widow's peak. So it's kind of annoying to write it all out, but you're going to have to write it, and usually they'll give you more space to do so. Okay, the next one that we're going to work on, let me draw a line of separation so that you can actually see how they're separate problems, since I already did a little bit of annotating on these problems. So the next thing we're going to talk about is sex-linked inheritance. So basically, sex-linked inheritance just means that it's about a gene that is carried on the X chromosome, meaning that males only get 1 copy, right? Because males are XY. Females still get 2 copies, just like any other chromosome. So males don't have a choice but to show the single allele that they got, right? There's nothing that can cover it up. There's no dominant or recessive. They just got what they got, and they have to show it. Okay, so if you look at this Punnett square, this is just a basic Punnett square for prediction of the gender of a kid, right? So if a mom can only give an X, she can only give an X in either case. A dad can give an X or a Y, right? So that means that 50% of their children are going to be XY, and 50% of their, each child born has a 50% chance of being a girl or a boy based on their sex chromosomes. So what is the chance that a pregnant female will give birth to a son? Well, the chance here based on this cross is that there's a 50% or there's a 1-to-1 ratio of XX to XY, which means that there's each child born has a 50% chance to be XY. And that's based on this cross that we did right here. Okay. So we're actually going to do a cross now that's kind of similar in setup to this one, but we're going to include an allele now that sits on the X. Okay. So a normal color vision male marries a female carrier. Color blindness is a gene on the X chromosome. And in reality, we tend to see a lot more males or men just being colorblind. And that's because if they get the colorblind allele, they don't have another X allele to cover it. So they're automatically colorblind. In the case of girls, because they get 2 alleles, they can get one that is colorblind and one that is not colorblind. And the non-colorblind one is dominant, so they'll just be a carrier. But their phenotype is that they can see normal color. It takes a lot more for a girl to be colorblind because both of their alleles have to, by chance, be the recessive colorblind allele version. Okay, so for colorblindness, we're going to write X with a capital N is going to be normal vision, and X with a lowercase n is going to count as colorblind. And remember, males cannot carry because they always show the one allele that they received. Okay, so we're going to have a normal color vision male marries a female carrier. Carrier means, again, that she has a normal one because her phenotype is normal, but her genotype contains a hidden recessive that she just doesn't have to display because it's not dominant. So we're going to have a cross between a normal vision male, so that's going to be normal on the X and nothing on the Y, crossed with a carrier female. So it's going to be a capital N and a lowercase n. And so we're going to write this out in our Punnett square. And notice again that I tend to draw them more horizontal rather than tall because drawing them tall does not help you squeeze in all of those letters, especially when you're doing a dihybrid cross like we did earlier. So here we're going to complete this and we're going to have capitals for the girls, another carrier for the girls, and then for the boys, for the boy offspring, we have to consider the ratio separate. Okay, so if you look at the bottom half, this is going to be for males only, and then if you look at the top half, it's going to be for females. And the reason I have it separated is because obviously these are XX and XX, and these ones are XY and XY. So you're going to take a ratio separate for males and separate for females, just because they're going to be considered differently because males only get one X. So what is the chance that these parents have a colorblind daughter? Now we're going to look here and we're going to look at just the top row for the females. Capital N, capital N, capital N, lowercase n. These phenotypes are going to be 100% normal, although there's a 50% chance that it's a carrier, right? So the chance that the parents have a colorblind daughter is actually a 0% chance, which is great news if you're looking for that. Now, the chances for a colorblind son. Well, we have a capital N here, so that's good. But over here we have a lowercase n, which means that there is a 50% chance of normal vision and 50% chance of color blindness. And that's because the mom is the one giving the X to the boy children because the dad already gave the Y to make the child a boy or a son. And the mom was a carrier, right? So that she can give either a capital N or a lowercase n, and it just by chance is a 50% chance of normal and a 50% chance of the son being colorblind. So that's what you have to take into consideration when you're working with sex-linked. The last one we're going to talk about is multiple alleles. And now this one is interesting because its case is kind of abnormal, but the rules still apply, just there's multiple rules at the same time. So here's an example. A man has a blood type A and his wife has blood type B. A physician types the blood of their 4 children and is amazed to find one of each of the 4 blood types among them. He's not familiar with genetics and calls upon you for an explanation. So we're going to use a Punnett square to provide that explanation. And blood types are often used to figure out, you know, in like TV shows like Maury, where they're like, you are not the father. It's because certain blood type combinations of two parents can only produce certain blood type combinations of their offspring. And obviously, if the kid's blood type doesn't match, then that means that somebody cheated. Okay, so for possible alleles in blood type, you have capital IA, capital IB, and lowercase I. These are the three possible alleles that blood types are made of. And I know there are three, but each time you do blood type, because you only have two chromosomes, you're still only going to get two copies at a time. So even though there are three options, you still only receive two at a time, one from your mom and one from your dad. IA and IB are codominant to each other. That means that when they show up, both the A blood type and the B blood type are made in the bloodstream, and so therefore that's a type AB blood. But both of them are dominant over the lowercase I. That's why they're written as capitals. So they're capitalized because they're dominant over the lowercase I. So if they are there, then the lowercase I doesn't show anything, right? Then only the blood type that is attached to the capital I is the main blood type, right? So the lowercase I is recessive to capital I alleles, regardless of whatever letter is on the capital I. So blood type A can be two versions. It can be capital A, or can be capital A and then lowercase I. Blood type B is similar. It can be capital IB, or it can be capital IB and lowercase I. Okay, the reason is because the blood that is created from the genes on the lowercase I, it has no markers on it, whereas the blood type B has a B marker and blood type A has an A marker. So you're still going to be making some blood type that doesn't have an A marker, but the type that you actually get recognized for is blood type B because your blood actually has the B markers present. It's different from not having any present markers, okay? Blood type AB, like I explained earlier, is having the codominance of blood type A and blood type B both present as IA and IB. And the last one is blood type O, which is lowercase I. And that's usually when you have a blood type O person marrying either an A or B that has a dominant and a recessive. You can also have a blood type O marrying a blood type O and producing kids as well. And that means that there's no markers on any of the blood. And blood type O was originally blood type 0 because there's 0 markers, but somebody misread it at some point and now it's considered O because the other ones are also letters. So a man is type A, a woman is type B. Is it possible to get children of type A, B, AB, and O blood from this cross? Well, we want to think about the possible combinations for type A. Remember, if the man is type A, that means his blood type is possibly capital IA, or it's capital IA and lowercase I. On the other hand, we have to think about the woman now too. Her blood type is type B, so she can be capital IB, or she can be capital IB and then lowercase I. Now, there are a bunch of combinations that we can try. I will list them out, but I'm only going to do the one that actually shows the answer, okay? So you can have all these different crosses. You can have capital A, and capital B. That's a possible cross. You can have this as a possible cross. You can have this as a possible cross. And you can have this as a cross. Okay, so there are 4 options because we're not exactly sure what their blood types are. We just know that it's A and it's B. Okay. So is it possible to get children of blood type AB, and O? Well, we're going to take a quick look at this cross right here. Okay, the reason I want to take a look at this cross is because there's a lowercase I and a lowercase I that have a chance to meet up. And remember, lowercase I plus lowercase I ends up being blood type O. That's why I'm curious to check this one out. So if we want to do this as a cross, we're going to do capital IA, I, capital IB, I, and we're going to produce a Punnett square out of this. And so we're going to have capital IA, capital IB, lowercase I, capital IA, lowercase I, and lowercase I. So here we have 1 IA, IB. Here we have 1 IB, I. Here we have 1 IAI. And here we have 1 that is little I, little I. That means this is blood type AB, this is blood type B, this is blood type A, and this is blood type O. So the answer to the question is, yeah, it's possible. Just because this is the possible combination of their blood type. If you have any of these other possible crosses, and they actually are confirmed to be one of these other 3 crosses for their blood type, then that means that either there was cheating, or it's just not possible to have this array of blood types in their offspring. So I'm going to ask that you go through and have worked on these all with me. If there were any questions that I didn't answer, you're responsible for answering them on your own. And I would love it if you could submit these notes for extra credit so I can see that you are following along.